My name is Eileen Carpenter. I am a physician scientist here at the University of Michigan in the Division of Gastroenterology. I joined as faculty in 2021, and I see patients with both benign and malignant diseases of the pancreas, and I also run a translational lab focused on pancreatic cancer. At the University of Michigan, we're very fortunate to have a dedicated multidisciplinary pancreas clinic that is focused specifically on patients with pancreatic disease. So I am really only focused on the pancreas, and it's really great because I get to interact with surgeons, oncologists, and we come together to formulate a very holistic plan for all of the patients that we see here. In establishing my lab, I wanted to contribute something to the field that I uniquely could do as a gastroenterologist that studies the pancreas. And so I decided to leverage the opportunity of endoscopic fine needle biopsy to develop a pipeline for my research. So we as gastroenterologists are uniquely positioned to aid in the cause of collecting biospecimens for pancreatic cancer, because we're often the first doctor that a patient might see with regards to the pancreas, because everyone needs a biopsy for tissue diagnosis. Now we're able to study all patients with pancreatic disease regardless of their stage. And so that's the basis of what I started my lab with. And since then, we have established over 50 pancreatic cancer organoid lines derived from patients from various locations. So does pancreatic cancer genetically change as it evolves within a patient? So the answer is yes. They've actually been doing studies like this for a very long time. The driver mutations are the same, but there is further evolution after the metastasis is established. And that is the main cause of what we call intertumoral heterogeneity within a patient. And that's how, tumors overcome chemoresistance, right? They keep mutating, they keep changing. And so if you responded to a certain therapy in the beginning, eventually you might not. This is a very important area of investigation in my lab, looking at the differences between the primary and the metastatic niche and trying to understand how pancreatic cancer overcomes drug treatment and develops chemoresistance. I had always known that I wanted to work on pancreatic cancer. My mom got diagnosed with stage 4 pancreatic cancer when I was 15, and she passed away not long after. In the end, there was no options for her other than palliative chemotherapy. And I think that's one thing to keep in mind, that, as physicians, telling a patient that they're out of options, that can be so heartbreaking. It was really a life-changing thing that I knew what I had to do. So we've got a couple projects going on. So one is looking at pancreatic cancer over time. I look at patients before they receive any treatment, and if it's possible to collect tissue after they've had treatment, then I collect that. And I've been using those matched biopsies. I've sequenced them and looked at the changes in the tumor before and after chemotherapy, and hopefully that can lend some insight on how chemotherapy alters the tumor and how the tumor can overcome chemotherapeutic killing. Another project is looking at pancreatic cancer over space, and I've already mentioned that a little bit about looking at the differences in the primary site versus the metastatic site. identifying what the differences are there and trying to understand how the tumor evolves as it goes from the pancreas to other sites. And then finally, I've generated several pancreatic cancer organoid lines that we can really use as patient avatars. So these all represent different patients. And we can use these to study different drug responses. And if a particular line is more resistant or more sensitive, we can do broad profiling to understand what makes that particular line more sensitive or more resistant, to understand how we can develop better precision pipelines for therapy. That's a great question. In the pancreatic cancer field, really what we need to do is we need to share our resources. And, I'm pleased to say that in the Nature Cancer paper we published back in 2020, we made that data broadly available. I worked together with Nina Steele on that. And that paper utilized the pipeline that I was telling you about with the endoscopic fine needle biopsies for single cell sequencing, and we've made that data set widely available, and many other labs have been able to use that data set and publish their own papers based on it, making the data widely available, allowing other researchers to query this data, especially in the area of bioinformatics and big data. This is more important than ever. I think as a researcher, you always want to see your work have direct clinical relevance. And so the next steps to this research is to identify potential therapeutic vulnerabilities and therapeutic targets and potentially open up a clinical trial based on that. I work in a multidisciplinary pancreas clinic, which affords me collaborations with oncology and with surgery. And we work very closely with an oncologist, Visa High, who is very involved in the clinical trials and runs several of his own. And so we collaborate together, and if a good idea shows up, then we can open up a clinical trial based on that. And that's how we advance research. The clinical trials are going to be what determine whether we can actually make a difference in pancreatic cancer patients or not. So I think we need to be doing more clinical trials, and we need to be more aggressive about that so that we can actually figure out if the stuff that we're doing at the bench makes any impact at the bedside. That's a great question. The other piece I'm going to say is that $1 million is not that much, but if I had maybe $1 billion, I'm just like. So I do think that to get something from the bench to the bedside requires a huge undertaking. A very huge undertaking. And here at the University of Michigan, we do have tools in place to make it a little bit easier, but still it requires going through a lot of red tape. It requires, a lot of regulatory, management. And I think it also requires, a lot of patient advocacy, because, patients don't really understand what clinical trials are about and how they actually help. And I would potentially open up maybe a translational bench to bedside sort of pipeline, but having one that's devoted solely to pancreatic cancer. And just bridging those gaps to make it easier for us to translate our bench findings into the clinic, I think would definitely be helpful to advancing the field and improving patient outcomes. So, for, the Sky Foundation, it's amazing that we have this resource in the Metro Detroit area. When we were going through my mom with pancreatic cancer, it would have definitely helped to have something like the Sky Foundation around us. But I became more familiar with the Sky Foundation when one of my close collaborators, Witch Kwan, who you'll be speaking with, approached me about collaborating for a project on pancreatic cysts. And so it was great that Sky Foundation has these seed grants, so we were able to apply for that and get it. And so through this collaboration with Dr. Rich Kwan, that is how I really got to know the Sky Foundation. The Sky Foundation funding, the grant on cysts We've been able to use some of those funds to pay for a study coordinator who we use to consent for our pancreatic cyst patients. So this is, an area that we can potentially intervene upon before pancreatic cancer develops. And we've also been able to use some of those funds to learn more about how to develop organoids for cysts, which is a little different than culturing organoids for pancreatic tumors. And then finally, we've also been able to use some of those funds to understand what are the specific perturbations in pancreatic cysts as they transform from benign to malignant. So the Sky Foundation to me is like a family. So whether it's providing a community or a support system, connecting survivors to each other and connecting patients to each other so they realize that they're not alone. And also, of course, the research funding where they provide seed grants to investigators with interesting ideas to try and advance the field.